Bismillahirrahmanirrahim. Welcome to all of you in today's lecture. In the previous lecture, I've started to give you an overview what is SAR and what is its relationship with the drug development. SAR means structure activity relationship, how it is useful, how it is adopted, how it is exploited for the drug development processes. In my previous lecture number 1 on SAR, I've explained you that any lead molecule, could be fine-tuned, through structural changes. A lead molecules could be fine-tuned through structural changes. You can call it structural modification. Okay, in order to control, in order to change its pharmacology. Pharmacological properties of a lead molecule, of a drug molecules, could be controlled, could be changed through structural modification, through structural changes. Those structural changes usually are minor ones. You do small minor structural changes on the lead molecule. As a result of smaller minor structural changes, the lead molecule change its pharmacokinetic behavior, its solubility behavior, its lipophilicity, its hydrophilicity. They undergo changes from minor to major changes in its pharmacological property. So in my previous lecture, I've shown you how a lead molecule could be used and how one can do different minor changes over that molecule in order to find the best one. You can find the best one, okay, the best having the best pharmacological activity. What is a lead molecule actually? I've told you over there. A lead molecule is that if you have, let's say, a library of 30 molecules, you have isolated them from certain natural source, or you have prepared them in laboratory, and you test those molecules for certain biological activity. Out of those 30 molecules, you find 4 molecules have shown a bit better pharmacological activity, biopotential. What you do, you take those 4, you look into their structures, you perform minor modification, you introduce hydroxyl groups or you introduce halogen groups, you look into the balance of whether it carries benzene group, benzene ring is there, you can expand the existing rings or you can reduce the size of the existing rings. Such sort of structural changes allow you to prepare other analogs. From one lead molecule you can prepare a number of analogs, and then you try those newly prepared analogs against for the same activity. Out of those prepared analogs, maybe 1 or 2 have the better activity than the previously starting lead molecule. Means you are doing fine tuning. Lead molecule has been used and you performed little structural modification and you test it again for the same activity. Maybe you will get some better results. If one gets the better results, that molecule which has some better result than the lead molecule, that has the capacity to become a drug in future. This is how you can play around with the structural modification. So keeping that SAR drug development topic forward, I'm going to explain it again with another example of a real drug molecule that is called amphetamine, you know. This drug has a very simple structure, so I can make a simpler explanation for you guys. This is called amphetamine. This is actually a psychostimulant. You may call it a CNS stimulant drug, you know. Okay, and it's a basic structure of this drug, you know. What people have done, they took this molecule and they played around its structure. They performed minor to major structural modification in order to prepare its analog, and they tried for the same, you know, CNS stimulant properties, and they see minor to major changes in its, you know, pharmacological activities. I'm just giving you a few examples what people have done, so you people could see how structural changes could give changes in its pharmacological activities. This is the actual molecule, you know, I've just shown. Few people came up, what they did, they prepared 3 analogs in the beginning, you know, just by doing methylation. They performed an alkylation, you know, reaction. You could see nothing special is done over here. This is the actual molecule. New analogs were prepared by simple methylation at the benzene ring, ortho, met and para. You know, the compounds were prepared and they were tested for their CNS stimulant activity. What they observed, this ortho substituted product, you know, of the amphetamine, it shows almost same activity as the original one. Okay, however, the meta and the para substituted amphetamines, they lost their activity. They lost their CNS stimulant activity. Okay, so you can see just a simple structural modification in the molecule change its pharmacological activity, change its pharmacodynamics, you know. Of course, the pharmacokinetics would have been changed as well. Then somebody came up again. Okay, this is the original molecule. Someone came with the preparation of this molecule. They produced oxidation of this carbon and this analog was prepared. Again, this analog showed similar effect compared to the original amphetamine, you know. So oxidation of the carbon give a new analog, a simple structural modifications. Then what the people did, you know, again an amphetamine was taken. Okay, they did side chain modifications. Sidechain modification. What was done? This side chain, this methyl group was extended, something like that. An extra methyl group was introduced over here. Okay. Similarly, another amphetamine was prepared over here. An extra methyl group was introduced at amine, at amine functionality. This was also kept in extension. So these two sort of analogs were prepared by somebody. This body, it lost its activity. It showed almost 1/10 activity potential of the original drug, So you see how much difference has been created by the introduction of the, by the introduction of minor side chain modifications. The pharmacological activity of this molecule was reduced also compared to the original one, but this body, which carried an extra methyl group at nitrogen, this lost the pharmacological activity considerably, and its activity was almost 1/10 of the original starting amphetamine. So just a little modification, I'm not showing you a sort of major structural changes. I'm just showing you methyl group on the ring and then methyl group on the side chain, Let me have you few more examples of the same drug. What the people have done, actually tried to fine tune the properties. They have tried to change, they have tried to introduce different substituents around the ring, around the side chain, so that they can control, they can find out what is the best, So you can see, you can follow what happened with that. This is amphetamine, again somebody came up and what they did, actually they closed this ring down, something like that. Okay, they prepared a new amphetamine having a carbocyclic ring over here. That is carbocyclic ring, and this body showed increase in activity compared to the original one. Okay, so activity of the ring was increased, activity of the amphetamine, this analog increased. It showed better activity compared to the one which does not carry ring, the original one. So this has happened as well. Then somebody came up again, people keep playing with the structure, Let me draw the original one again. This is the original amphetamine, and from here this amphetamine with carbocycle was prepared. Then somebody again came up and used this body again. Okay, what he did, they keep the cycle, but they introduce unsaturation over here. One double bond was introduced in the ring, A new analog was prepared. Over here, when from amphetamine a ring was introduced, its activity increased compared to this original one. However, once somebody moved one step ahead, introduced one unsaturation, this compound, interestingly, lost or reduced its activity and showed almost half of the activity compared to the original one. Could you see? when this cycle a ring was introduced, the introduction of the ring increased the activity. However, in the same ring, the introduction of unsaturation that reduced the activity almost to the half of the original one. Structural changes, minor structural changes. They are so-called minor structural changes. In fact, they are not the minor changes as far as the pharmacology or the pharmacokinetic is concerned. Actually, these smaller structural changes cause a significant changes in pharmacokinetics, which results in change in minor to major changes in pharmacodynamic properties of the drugs of the lead molecules. So these are the few of the examples again. I'm just trying to convey you people that how simple changes are actually causing so many differences in pharmacological effects. Then again, people use this body. Nothing special. Initially, somebody introduced halogens. A chloro group was inserted at para position. Okay. Then somebody again produce another group where CF3 was introduced in the ring. Then somebody came up again where methoxy group was introduced. So this is how people keep on playing, and then they looked into their pharmacological activities as well. Then somebody again came up, instead of this oxygen, they also prepared another type of amphetamine. Over here sulfur was used, SCH3 was introduced. So different substituents were introduced again into the ring, and their pharmacological activities was compared with the original drug molecule, and considerable increase and decrease in the pharmacological actions were observed. Again, I'm just giving you an example again, just to wind it up now. Okay, then people came up, they started to play around this area. Okay, they produce this sort of amphetamines, a major class of amphetamines, known for having such sort of structures. They started off from amphetamine. I could do that again for you, so keep you on track. Is that okay? I can write over here. This is I'm trying to write the regional amphetamine. Okay, so you keep that in mind. This was built over here. Then people again they introduce methyl groups over here as well. Okay, just to create new components from here. In another body, what they did, they keep only one methyl group at this point, So this is how another class of amphetamines was built, was created to study the pharmacological actions of these new analogues, and their properties were compared as well. So this is what actually has been done with a single simple molecule having very simpler structure, Let me recall again one more example actually. Few people started to play around this area. Okay, initially introduction of the chain or introduction of the uh, substituent was done on this side. Next, people use these two carbons. Okay, they build 5-membered ring over here. Then different substituents was also introduced to prepare different analogs as well. So another class of amphetamine was developed having 5-membered rings over here with the heteroatoms. So this is all about amphetamine. This is what I'm trying to say. If a lead molecule shows a promising pharmacological activity, you can play around, you can introduce different groups, in order to find the best molecule. Okay. Similarly, an existing drug like amphetamine, through R&D, research and development, you can fine tune that, you can enhance its activity, you can produce its new formula, you can introduce new structural modification into the drug to increase to control its activity. Whether you have an interest to increase its like lipophilicity, sometimes you have an interest to increase its excretion from the body. So similar sort of structural changes could be made so you can control the pharmacological actions of the drugs inside the human body, by playing around with the structural modifications. Let me rewrite again amphetamine and try to recall it for you. What we did initially, we introduced different substituents, simple methyl groups here. Then somebody came to introduce structural modification here, sidechain modification over here. Then someone came up with introduction of the carbonyl functionality over there. Then some people came up with the closure of the ring, Okay. Then one man came up with the introduction of the unsaturation in the closed ring. Then again, few of the people came up, okay, with side chain introduction in this area. Then few of the people came up and they introduced side chains on this area. Okay. So whatever we are doing, whatever they have been done, let's say a CH3 has been introduced over here. Where it has been introduced, they have introduced in amphetamine. Okay, they have introduced over here. Similarly, they use this molecule again, amphetamine, starting from here. They have introduced over here, or they have introduced over here, or in next they have oxidized this carbon. Whatever has been done from here to here, you could see what is it? Introduction of certain substituents. Being chemist, tell me what is the who is going to do such structural changes? You being the pharmacist, pharmaceutical chemist, you people are going to do that. You need to know if you are going to introduce a methyl group at the ring, how that introduction will be done, what sort of the reaction you need to do that you can introduce a methyl at ortho position. Then when you want to introduce the methyl at meta or para position, what sort of the reaction should be done, what will be the reagent, what will be the conditions. Similarly, if you are going to oxidize the carbon, benzylic carbon, you are going to make it a carbonyl functionality. What sort of the reaction has to be done? So what is I'm trying to say? the drug development, the SAR, structure activity relationship, and the drug development that purely relies on your concepts in organic chemistry. You need to have an idea what sort of the reaction has to be done, how has to be done, how the reaction has to be controlled, you know, when you are going to insert, when you are going to introduce a substituent of your own choice, you know. A methyl is going to be introduced at the ring. If the methyl has to be introduced at the amine functionality, your requirement, your reaction criteria, your reaction conditions, your approach is supposed to be entirely changed, you know. Okay, you agree or not? You have to agree actually. Similarly, if you're going to do a oxidation of this carbon, you need to have certain reaction parameters, certain reaction conditions, certain reagents. So what those will be mean you need to have strong knowledge in your basic chemistry related to the reactions, nucleophilic substitutions, electrophilic substitutions, oxidations, reductions, you know. Basic reactions in organic chemistry are required once you are interested in SAR drug development, you know. You need to have a strong grip over how the reactions are controlled, how the reactions are performed, and how you can introduce the substituents of your own choice at your point of your choice as well. So this is all about in today's lecture about SAR, SAR, structure activity drug development. At the end, what my message is for you guys: a pharmaceutical student should have a strong background in organic chemistry so he can exploit lead molecule, drug molecule to find, to develop the best ones, you know, with best pharmacological activities. Thank you very much. If you have any questions, don't hesitate to contact me. I'm always available. Thank you ji. Assalamu alaikum.